Hello viewers, welcome to our channel. In this video, we will discuss some important questions on enzyme. This is very important topic. For CSR NEET, GATE, NEET exam, even for BSc MSc student, the questions regarding this topic is very important. Some brief notes regarding the questions and their related options are also provided here, which will be beneficial for you. So stay tuned in this video. The first question. Enzyme acts best at a particular temperature called option 1 minimum temperature, option 2 maximum temperature, option 3 optimum temperature and option 4 none of the above. And the correct answer is option 3, that is optimum temperature. The ideal temperature for enzyme is 20 degree to 35 degrees centigrade. At very low temperatures, they become inactivated, and at very high temperature, like higher than 45 degrees centigrade, they get denatured. So the enzymes get inactivated at low temperature, whereas they are denatured at high temperature. The low molecular weight enzymes are more heat stable than the high molecular weight enzymes. Next questions. Enzymes, vitamins and hormones are common in option 1 being proteinaceous, option 2 being synthesized in the human body, option 3 enhancing oxidative metabolisms. Now option 4 is regulating metabolism, and the correct answer is option 4, that is regulating metabolism. Now the first option is that being proteinaceous. Enzymes are proteinaceous, but vitamins are not proteinaceous because proteins are macronutrient, whereas the vitamins are micronutrient. So this first option is not correct. Okay. Even the hormones are not always proteinaceous. Hormones may be either protein or steroid. So this first option is incorrect. Next option is being synthesized in the human body. Enzymes are synthesized in the human body, but not all the vitamins are synthesized in the human body. Vitamins D can be synthesized in the human body, but not vitamin A, B, etc. are not synthesized in the human body. Enhances oxidative, so it is also incorrect. Enhances oxidative metabolism. This option is also incorrect for enzymes, vitamins and hormones. Both all are not involved in enhancing oxidative metabolisms. But enzymes, vitamins and hormones all are helps in regulating the metabolisms. Now let's discuss about some characteristics of enzymes. Enzymes are complex macromolecules with high molecular weight. They catalyze biochemical reaction in a cell. Enzymes do not start a reaction. Enzymes affect the rate of biochemical reaction, not the direction of the reaction. Enzymes are specific in reaction. Enzyme activity decreases with increase in temperature. Next question. The enzyme glucose 6-phosphatase is present in option 1 liver, option 2 muscle, option 3 adipose tissue and option 4 is brain. And the correct answer is option 1 liver. So the enzyme glucose 6-phosphatase is present in the liver. Now let's discuss about this enzyme. The enzyme glucose 6-phosphatase catalyzes the hydrolysis of glucose 6-phosphate, resulting in the creation of phosphate group and free glucose. The reaction is like that: glucose 6-phosphate plus H2, it produces D-glucose and phosphate with the help of the enzyme glucose 6-phosphatase. Phosphatase. This phosphatase break the phosphate group, hydrolyze the phosphate group, okay, and release this phosphate group. In case of glucose 6-phosphatase, it detach this phosphate group from the glucose and produce phosphate and free glucose. And after this reaction, when the free D-glucose is produced, The glucose is then exported from the cell via glucose transporter membrane proteins. Next question. Factors affecting enzyme activity is option 1 concentration, option 2 pH, option 3 temperature, and option 4 is all of the above. And the correct answer is option 4, that is all of the above. Enzyme activity can be affected by a variety of factors such as temperature, pH, and concentration. Enzymes work best within specific temperature and pH ranges, and suboptimal conditions can cause an enzyme to lose its ability to bind to a substrate. The pH optima of most enzyme is option 1 between 2 and 4, option 2 between 5 and 9, option 3 between 8 and 12, and option 4 below 1. And the correct answer is option 2, that is between 5 and 9. In general, an enzyme has an optimum pH. Although most enzymes remain high activity in the pH range between 6 and 8. Some specific enzymes work well only in extremely acidic or alkaline condition. Next question. The Koshland's theory of enzyme action is known as option 1 lock and key model, option 2 induced feed theory, option 3 enzyme substrate complex theory, option 4 none of the above. And the correct answer is option 2, that is induced feed theory. Now let's discuss about this Koshland theory of enzyme action. According to the induced feed theory by Daniel Koshland, binding of a substrate to the enzyme brings a conformational change in the enzyme to either enhance or inhibit its activity. The active site keeps changing in its shape until bound to its substrate. The final binding gives proper shift to the enzyme. Whereas in case of the lock and key model, the shape of the active site or the enzyme is not changed. It remained fixed. So that's in the case of lock and key model, the shape of the enzyme remains fixed. Whereas in case of the induced feed theory of Koshland theory or induced feed theory, the shape of the active site on the enzyme is keep changing. Okay. Next question. The enzymes involved in feedback inhibition are called option 1 enzyme, option 2 coenzyme, option 3 allosteric enzyme, and option 4 is apoenzyme. And the correct answer is option 2, that is allosteric enzymes. Whereas the apoenzyme is the protein part of an enzyme, holoenzyme is protein plus non-protein plus of an enzyme, and coenzyme is the non-protein part of an enzyme. Allosteric enzyme is an enzyme that contains a region to which small regulatory molecules, that is called the effectors, may bind in addition to and separate from the substrate binding site and thereby affect the catalytic activity. Next question. The term enzyme was coined by option 1 Fischer, option 2 Koshland, option 3 Kuhn, and option 4 is Arrhenius. And the correct answer is option 3 Kuhn. Next question. The enzyme which helps in the formation of peptide bond is known as option 1 peptidase, option 2 peptidyl transferase, option 3 carbonic anhydrase, and option 4 is none of the above. And the correct answer is option 2, that is peptidyl transferase. The peptidyl transferase is an amino acyl transferase. So its primary enzymatic function to the ribosome, which forms peptide bond between adjacent amino acids using tRNAs during translation process of protein synthesis. So the enzyme class of this peptidyl transferase is aminotransferase. Its primary function of the ribosome is to form the peptide bond between adjacent amino acids using tRNAs during translation process of protein synthesis. During translation process, amino acids are sequentially added, and these amino acids are joined by the peptide bond. And this peptide bond is formed with the help of this peptidyl transferase. Whereas the enzyme peptidase keep the peptide bond. Okay, AS means peptidase. Okay. Next question. Allosteric enzyme possess option 1 active site plus allosteric site, option 2 active site plus 2 types of allosteric sites, option 3 active site plus 3 types of allosteric sites, option 4 only 2 types of allosteric sites, and the correct answer is option 1, that is active site plus allosteric site. Allosteric enzymes do not need to be oligomers as before, and several systems have actually shown an allosterically inside a single enzyme. In biochemistry, the regulation of a protein by binding an affected molecule to the site other than the active site of an enzyme is known as allosteric regulation. So the site where the enzyme binds other than the active site is known as this allosteric site. Next question. Zymogen is option 1 enzyme precursor, option 2 enzyme inhibitor, option 3 enzyme modulator, and option 4 is enzyme precursor. And the correct answer is option 1 enzyme precursor. Enzyme precursors are called zymogens or proenzymes. Examples are enzymes of the digestive tract in humans. Some protein precursors are secreted from the cell. Many of these are synthesized with an N-terminal signal peptide that targets them for secretion. Next question. Which enzyme is true for enzyme? Option 1 do not require activation energy. Option 2 act by lowering the activation energy. Option 3 increase requirement of activation energy, and option 4 none of the above. And the correct answer is option 2 act by lowering the activation energy. Enzymes generally lower activation energy by reducing the energy needed for reactants to come together and react. Next question. The first trace enzyme is option 1 carbonic anhydrase, option 2 DNA gyrase, option 3 DNA polymerase, and option 4 is trypsin. And the correct answer is option 1. The first trace enzyme is carbonic anhydrase. Carbonic anhydrase catalyzes the reversible reaction between carbon dioxide and water to form carbonic acid. Enzymes are actually composed of option 1 carbohydrate, option 2 proteins, option 3 fat, and option 4 is vitamins. And the correct answer is option 2 proteins. Enzymes are protein composed of amino acids linked together in one or more polypeptide chains. The sequence of amino acids in a polypeptide chain are called the primary structure. This in turn determines the three-dimensional structure of the enzyme, including the shape of the active site. In competitive inhibition, Km increased but Vmax unaffected. Option 2, Km decreased but Vmax unaffected. Option 3, both Km and Vmax decreased. And 4 is Vmax decreased but Km unaffected. And the correct answer is in competitive inhibition, Km increased but the Vmax unaffected. So, if you draw this line, then it is 1 by V, it is 1 by S. for inhibitor, and for this line it is no inhibitor. Okay, and this is 1 by Vmax. This line is 1 by Vmax. Incompetitive inhibition competes with a substrate for binding to an active site. When the inhibitor occupies the active site, it forms an enzyme inhibitor complex. Next question. In non-competitive inhibition, option 1, Km increased but Vmax unaffected. Option 2, Km decreased but Vmax unaffected. Option 3, both Km and Vmax decreased. Option 4, Vmax decreased but Km unaffected. And the correct answer is option 4, that is Vmax decreased but Km unaffected. I just try to draw. This line is for, it is for inhibitor. And this line is for no inhibitor. Okay. This is 1 by B. It is 1 by So here the inhibitor Vmax decreased with inhibition, with no inhibitor, with inhibitor. So Vmax is decreased. Okay, but Km, this Km is unaffected. Okay, Vmax Km same. In inhibition, no inhibition, Km remains same. But Vmax, it is with inhibitor the Vmax is there. Vmax is decreased. Okay. In non-competitive inhibition, a type of allosteric regulation is a specific type of enzyme inhibition characterized by an inhibitor binding to an allosteric site, resulting in decreased efficacy of the enzyme. An allosteric site is simply a site that differs from the active site where the substrate binds. Next question. In uncompetitive inhibition, option 1, Km increased but Vmax unaffected. Option 2, Km decreased but Vmax unaffected. Option 3, both Km and Vmax decreased. Option 4, Vmax decreased but Km unaffected. And the correct answer is option 3, that is both Km and Vmax decreased. So, just remember that in competitive inhibition, Km increased but Vmax unaffected. Whereas in non-competitive inhibition, the Km affected but Km unaffected and Vmax reduced. Whereas in uncompetitive inhibition, both Vmax and Km decreased. Okay. This competitive inhibition, non-competitive inhibition, this related question is very important. Just remember. So just try to draw this. This is for uncompetitive inhibition. Okay. Line. This is 1 by S. This for substrate concentration. This is 1 by V for LS for T. And this one is, first one is inhibitor. This one is no inhibitor. No inhibitor. Okay. So here, just see that this is the Vmax. For no inhibitor, this is Vmax. For with inhibitor, this is the Vmax. So the Vmax decreased, whereas it is the, this is the Km. The Km here is Km also. This is the Km for inhibitor. This is Km for non-inhibitor. So Km also decreased. So Km decreased and Vmax is also decreased. So in uncompetitive inhibition, both Vmax and Km decreased. Okay. So in uncompetitive inhibitor, both Km and Vmax decreased. Whereas in non-competitive inhibition, only the Vmax decreased. Whereas in competitive inhibitor, only the Km increased, but not the Vmax is unaffected. Okay. Now, in uncompetitive inhibition, also known as anti-competitive inhibition, takes place when an enzyme inhibitor binds only the complex formed between the enzyme and the substrate, that is the ES complex, enzyme substrate complex. Uncompetitive inhibition typically occurs in reaction with two or more substrates or products. This is all about the MCQs on enzymes. If you like this video, please like, share and subscribe to this channel. Thank you.